Welcome everybody to our monthly seminar series. We are just waiting, a few seconds so everybody is able to join us. And then before to start with our trainee talk today, I have some few announcement, but we are just waiting a little bit more. Welcome everybody again to the monthly trainee talk of the Cancer Cachexia Society. Today we have the trainee talk, but before to start, we have some announcement. First, we want to announce a new activity of the Cancer Cachexia Society, that is the Clinical Trials Advisory Board. This will be an independent expert review to strengthen your clinical research. What they will do a confidential, independent, comprehensive review supporting funding and development decision, or a lot of type of clinical trial, observational study, biomedical development, and quality cachexia research. For all the information that you want, please go to check in our website, www.cancercachexiasociety.org. I want to remind everyone that our next seminar series will be on May 13, and that day we will have Revati Dewal, that is a senior editor of Nature Metabolism, and also we will have Maria Garcia Fernandez, that is a senior editor of Nature Communication, and this talk is called Meet the Editors, and they will, talk about an editorial process that taking place in the Nature portfolio. Finally, today we have three fantastic speakers that are our trainee, and we will have three. The first that will speak is Noah James Forrest from Northwestern University, and he will talk about unsupervised machine learning approaches for classifying cancer cachexia, cancer-associated cachexia. The second one will be Chiara Ninfali from the Institute for Bioengineering of Catalonia, functional assessment of cancer cachexia using 3D tumor muscle model. And then the last one will be Abolfazl Nik-Akhtar from University of Ottawa, and he will talk about reciprocal multi-omics reveals that modulation of ADP-ribosylation in skeletal muscle shape LLC-induced cancer cachexia. I just want to say that this time is a little different. They have 15 minutes for their presentation, and then in the middle, after that, each of them, they have 5 minutes for question. So now I will stop to share my screen, and I please ask to Noah to start to share his screen.
Good. Thank you for the introduction, Fabrizio. I'm really honored to be speaking here today. I defended my PhD last spring, about a year ago exactly today, and I've been in clinical environments for the last year and kind of seeing a lot of cachexia from the clinical perspective, so I'm really recognizing the importance of this talk. So I'm looking forward to other speakers talking as well. So yeah, my name is Noah. I am at Northwestern in Chicago in the US. And I'm going to be talking about unsupervised machine learning approaches for classifying cancer cachexia today. So I'm sort of preaching to the choir today because I know we're all very interested in cachexia, but something I just want to keep in mind when thinking about this syndrome is that cachexia is really this multifaceted disease that consists of several different components. One that I'll highlight and that I'll talk about to a certain degree is the inflammatory cytokines that really start a lot of the downstream processes in cachexia that lead to the muscle wasting phenotype that we see so often in clinical environments. There are other components in the central nervous system resulting in decreased appetite, and really the end result is what we observe in the clinical environment, which is weight loss and, more importantly, the muscle wasting. So I'm just keeping that in mind, that there are many different components to this syndrome, and it's something that I want to try to capture in clinical data. Despite knowing that cancer cachexia consists of these several different components. A lot of the things that we use clinically, a lot of the tools that we use, categorize and grade cachexia severity on like a one-dimensional scale. So the most famous and widely used one, even still, is the Fearon consensus criteria from originally from 2011, and then some spin-offs from 2015, the WLGS, which I'll talk about. And really, this divides patients into cachexia severity based off of severity of weight loss. Obviously, the WLGS takes the BMI into account as well. So we have a syndrome that's multidimensional, but in clinical practice, we're really only using unidimensional scales. And even newer systems that kind of take lab markers, biomarkers into account really only give patients like a less severity or more severity. So I thought that there would be a need for designing a system that categorized patients in a multidimensional fashion. And because I'm been in the clinical environment now for the last year, I like to think about how my project kind of applies to patients with different cancer types in which cachexia is prevalent. So here's one, a common one that I saw in the Surg-Onc service and the cardiothoracic service. It's a man in his 60s who's recently diagnosed with non-small cell lung cancer. He's had like a fairly stable weight over the last few months, like less than 2% of total body weight loss. But like on initial laboratory testing, we noticed like an absolute neutrophil count that was slightly elevated and an absolute lymphocyte count that was mildly depressed. So it's like something I was thinking of, and questions I think about this patient are, do the current clinical systems that we use to categorize cachexia, is this patient considered cachectic yet? And I would say no, he's not really lost any weight, probably not showing many signs of emaciation. But would these lab markers indicate like a poor outcome, poor survival? Does he have higher risk of disability down the line and being hospitalized? So a primary objective, as I've kind of alluded to, is to design a clinical cachexia classification system that reflects the multidimensional nature of the disease that's well known in preclinical models. And the hypotheses that are sort of driving this objective is that cachexia manifests and displays itself in different ways in human populations and across different disease dimensions. And then the final one, which is like more getting to the clinical utility, is that if a system that's used clinically reflects the syndrome's underlying multidimensionality, maybe it does better at predicting patient outcomes than a unidimensional system that's currently in place. So I did this analysis early in grad school, and I kind of put it on the shelf for a long time because I was just working on other stuff. This is in a 1,500 patient cohort with non-small cell lung cancer treated with immune checkpoint inhibitor therapy. And I just kind of did this sort of out of curiosity. I took four variables, their percent weight change, absolute lymphocyte count, and neutrophil count, as well as their BMI. And I just threw it into a k-means clustering algorithm, like very simple. And I found some like very interesting finding, most notably of which is like this group in this PCA plot of the orange dots. They had the worst survival out of the four subgroups that were discovered in the clustering algorithm, but they had no overt signs of weight loss clinically. All that they displayed was an absolute neutrophil count that was elevated. So we kind of called this the occult inflammatory phenotype of cachexia. They haven't lost any weight, but they're showing some signs of maybe acute inflammatory processes that might lead to poor outcomes down the line. And again, I put this on the shelf, not really thinking much of it. I used it as some preliminary data in a grant application. But then I thought for my dissertation, I wanted to expand upon this. So before I did that, I wanted to develop this multidimensional system in a larger cancer cohort that included many different cancer types. So I used a data source that's called the NMEDW here at Northwestern. It's the back end of our electronic medical record. And I won't belabor this point. I spent a lot of time on this point, like making sure I had a really high quality cohort for downstream analyses. But essentially, I ended up identifying approximately 7,500 patients with non-small cell lung cancer, pancreatic cancer, and colorectal cancers with enough data to analyze. And one of the limitations of that preliminary analysis that I showed you in that k-means clustering is I had to throw out a lot of patients that were missing data. As some of you may know that work with clinical data, especially electronic medical record data, a lot of data is incomplete. For whatever reason, patients may come from other institutions and may not have complete data files when they come to Northwestern. Perhaps some of you that live outside of the US have more complete hospital records. That's like my dream. But at least in the US, when someone comes to Northwestern, not all of their data from other hospitals gets shared. So I needed to implement a system that accounted for the missing data. And something that I found in the literature that had never really been applied to real world data, it was kind of like a thought experiment and by a statistician, was this multiply imputed cluster analysis that occurs in four phases. Essentially, you take some variables, in this case, like cachexia associated variables from prior literature, and you create many number of imputed data sets. You then cluster each of those data sets, and then take the centers out of those clusters and then cluster those centers again. And then you have like this sort of rudimentary voting system where a patient ends up in a final cluster based off of the number of times that they were assigned to a particular center. So it uses imputation as well as like several iterations of clustering to assign a final cluster to a patient. And so you might be asking, like, what data did you include in this clustering algorithm? I did it three different ways for the purpose of my dissertation. I'm an informatician at heart, so data is like a big passion of mine, as well as the people that were reviewing my dissertation. So I used three different data sets. I'll focus on this data set one primarily today, just for the purpose of time. But this one included known clinical biomarkers of cancer cachexia. So I mentioned absolute neutrophil count. You might be thinking neutrophil to lymphocyte ratio, which was included in this, as well as some weight loss associated variables and other components. So here they are in this data set one, kind of divided into their broader categories. So the weight loss grading scale, which is well-validated weight loss associated metric in cancer cachexia, some serum markers of malnutrition, most notably the prognostic nutritional index and the hemoglobin, and then some physical function metrics, most notably the ECOG scale, and then markers of inflammation, which was captured by the neutrophil to lymphocyte ratio. And then with this data set one, after the several iterations of clustering in the MICA pipeline, found seven different clusters that had different rates of survival at 1 year. So starting at cluster one, this is arranged in order of best surviving clusters to worst surviving. At 1 year, cluster one had about 88% of the patients surviving, and cluster seven had 63%. So at the next step was kind of understanding, well, what types of patients are included in each one of these clusters. Are they meaningful? Are these clusters even interpretable? So I used RPART, like a partitioning algorithm, to sort of parse out what variables were most important in defining each one of these subgroups. And I really wanted to highlight cluster six. It's like most relevant to the patient case that I presented towards the beginning of the presentation. And they were defined by a very high neutrophil to lymphocyte ratio, again, without any overt signs of weight loss. So very similar to that preliminary analysis that I did in the non-small cell lung cancer cohort treated with immunotherapy. And they had notably worse survival. If I go back, cluster six, only 70% of these patients were surviving at 1 year post-cancer diagnosis. And then cluster seven had a bunch of abnormal, the worst surviving cluster had a bunch of abnormal serum markers as well as clinically overt weight loss. So it appears to be additive. And getting at one of the central hypotheses of this project, does this multidimensional classification system do better head-to-head when compared with a existing cachexia classification system, in this case, the WLGS? So on the y-axis here, you have the area under the receiver operating curve, like a measure of accuracy in predicting overall survival. And I did it at several different time points. And as you'll note, the black box here, when compared to the WLGS in blue, did significantly better per the AORUC metric at predicting whether or not a patient would be surviving at each one of those time points. So this is not a perfect way to test. This is like a sort of preliminary way of testing. The only reason I say that is because, these clusters were designed with this cohort in mind. So there is definitely some bias here because the unsupervised clustering was done on the cohort. Currently working on applying these clusters to other cancer cohorts. But this was like a really great signal telling sort of telling me and my collaborators, this might be a good way of thinking about cachexia. There might be this group of patients, cluster 6, that inflammatory, occult inflammatory subphenotype that might actually have some clinical relevance to it. And then getting into some secondary outcomes, that was in predicting overall survival in the last slide. But I was also really curious about disability-free survival because cachexia, as we all know, contributes to poor overall survival, also carries a large morbidity to it. So this was showing stratification of the different clusters in terms of their disability-free survival, which is defined as time without physical therapy or occupational therapy ordered, as well as inpatient physical therapy. And you'll note that cluster 6 in red, again, that's the occult inflammatory subphenotype that I've been referring to, had actually the quickest time on average to having a PT or OT ordered for them, sort of suggesting that there was a significant amount of morbidity related to this subphenotype of cachexia. And then again, another outcome we wanted to look at is, how severe are these patients' disease? And this is sort of indirectly captured by hospitalization-free survival. In panel A on the left, this is the WLGS in different categories stratified, and there was actually no difference between hospitalization between different WLGS categories. But when in panel B, when you stratified by the subphenotypes discovered through the MICA pipeline, we saw significant differences in the strata. So again, kind of just thinking about burden of disease as well as morbidity. So some conclusions from this analysis, using that R partitioning model, we were able to interpret what these clusters meant from a more clinical interpretable perspective. And then they're like really one of the central hypotheses of this project was can some dimensions of cachexia, in this case acute systemic inflammation captured in the neutrophil lymphocyte ratio, they can sometimes manifest independently of others, such as weight loss or malnutrition in some subpopulations of patients. And then some of the performance metrics, the clustering system performed better at predicting overall survival compared to some existing systems. And then as it detected some significant differences in time to event for the secondary outcomes, most notably disability-free survival, as well as hospitalization-free survival. I'm interested in urology. That's what I'm applying into for residency. So I'm currently working with some urology faculty here at Northwestern, trying to apply this clustering system to some urologic malignancies, most notably urothelial cancer, so bladder cancer, as well as prostate cancer. So stay tuned for kind of, the external generalizability of this system as well. And that is all I have.
Thank you very much, Noah, for this fantastic talk. I want to tell that everybody can insert answer in the Q&A and I will read them. So we have a first question, and beautiful work and great work. I was wondering if in cluster 6 for inflammatory markers, apart from IL-6, what other markers did you identify that are highly relevant to cachexia development in these patients?
Yeah, that's a great question. And so I was originally looking at IL-6, but I excluded it actually from the clustering algorithm because it's not routinely measured in clinical settings. So really part of this system is not only one capturing distinct subgroups, but something that using variables that are routinely clinically captured. I would love to get IL-6 data, TNF alpha data would be amazing, but it's not something that's routinely ordered, at least in the US, for measuring inflammation. So neutrophil to lymphocyte ratio is kind of one of the better markers we have at this point.
Another question is very interesting work and is similar to the work that we did in Glasgow. We tested the use of CRP and MGPS to
Yeah.
assess inflammation. We found this to be superior prognostically to NLR. Did your cohort not have CRP available? How did you determine to cut off an NLR or 6?
Yeah, an LR of 6, semi subjective. It was really based off of one of my dissertation committee members, who's Ishan Roy, who's a cachexia researcher. He suggested 6. Me with zero clinical context, I actually didn't know why this cutoff of 6 was recommended. Towards the question of CRP, initially was included in the clustering algorithm. Unfortunately, at least in the US, it's maybe a practice difference in different countries. In adult populations, CRP is not routinely measured in cancer populations. I think it should be. It sounds like I don't see who asked that question, but it sounds like it could be a very valuable prognostic marker. But at least in our data set, it was so infrequently measured that it didn't do well in the imputation phase. So we actually excluded it from the clustering. But a great question. I think it's something we could make clinical recommendations for in the future.
Very cool work. Have you noticed membership differences between cluster depending of the patient demographic?
Yes, great question. I could talk about this for a long time. Patients tended to be older in the poorer surviving clusters, but age was not a variable that we included in clustering. So it suggests perhaps there's like a sarcopenia component to this, and I think it warrants some further investigation. But yeah, that was one of the demographic differences that stood out the most.
I think we have time for a last question. So also, are there plans to include an objective measurement of functions such as n-grip strand?
Yeah, these are great questions. I love talking to cachexia researchers. So again, we were using like a real world clinical data set. Ideally, we would love to use a functional measure such as grip strength because I know that's like the gold standard. However, when you're using this real world clinical data set, you use the best that you have. So we sort of came up with these markers such as disability free, which has been highly cited in the cachexia space as sort of a surrogate marker for function. But yeah, I think it would be great to apply these clusters to patients in a research setting and see if they correlate to things like grip strength. Yeah.
Thank you so much. I'm sorry we cannot answer the other question, but please feel free to send all the question to Noah email. That isn't. Thank you so much, Noah.
Thank you.
Please, Chiara, you can share your screen. Can you? Yes. You mute. You are mute. You should be at the left, bottom left of your screen.
Okay, now can you hear me? Yeah?
Yes, but please share again your screen.
Yeah. One moment. Okay, can you see and hear now?
Yes, we can. Thank you.
Okay, good afternoon. I'm Chiara Ninfali from the Institute for Bioengineering of Catalonia here in Spain. And today I will show you the very first results of a new project about the assessment of cancer cachexia from a functional point of view using a 3D tumor muscle model. So a really short overview about the conventional models to study cancer cachexia that of course are 2D cultures. So we can seed myoblast in a plate, we let them differentiate for several days, and then we can treat them with cancer conditioned media and then analyze them with a microscope or molecular point of view. Of course, this model works, but it has the limitation that we have a plate that has a stiffness, and we have cells in 2D over it that are randomly organized. They form myotubes, but randomly. The other model, of course, is the mouse model that is more complex, where we have a tumor inside a mouse that can be genetically induced or inoculated. But we have to wait several days to obtain a phenotype, a cachectic mouse, and then we have to sacrifice this mouse to do muscle analysis. This model works, is more complex, but of course we have ethical concerns due to the fact that we use animals, and also we have animals in our end that is not a human model. So in the last few years, it has been developed this technology that is named organ on a chip, where we can combine microfluidics and 3D cell culture on microchips. We have the microfluidics that combines 3D cultures. This is fine because it mimics human organ physiology and function in vitro. It enables realistic disease modeling and drug testing. It supports personalized medicine approaches because we can use patient-derived cells. Of course, it reduces reliance on animal testing, and it permits faster, cheaper, and more ethical drug development. So the objective of this new project is to build a 3D engineered system to model human cancer cachexia, also assessing muscle function from a functional point of view. For people that we work in muscle, we know that for muscle tissue, shape matters because structure is essential for muscle function. It's not the same to have a plate where we have some myotubes inside in a monolayer, than having highly organized, aligned fibers that form multinucleated cells that together form a functional unit that is named sarcomer. How can we reach this objective with tissue engineering? So in our laboratory, we use 3D printer to design these molds that are made of PDMS, it's like a silicone, and they are formed by inner chamber here that have two pillars that are flexible. Here is the top view and the side view. How can we use these molds? We can encapsulate, we call it encapsulate, patient-derived myoblasts, so myoblasts that derive from humans, from patients, with a fibrin matrigel matrix. They form a matrix and we put together in this chamber. This is how they appear the day of encapsulation. So we let them proliferate for 2 days when we put differentiation media. As you can see here, after 2 days, the matrix is compacted and they are around these two pillars. So we let them differentiate for 18 days, and these two pillars gives the tissue a continuous tension that helps to orientate muscle fibers. So finally, we will have myoblasts that are aligned around the pillars, that they are fused, and that they are functional. This is how they appear after 18 days. So if we cut a longitudinal section, we can see that our fibers are all aligned. This is sarcomeric alpha-actinin and F-actin in red, and this is the merge. And if we cut in a transverse section, we can see that the muscle is completely has a lot of fibers inside. Okay, we have aligned fibers, so we want them to function, to contract. How we can do it? We can put 2 electrodes, graphite electrodes, to the side of the muscle, and we can apply an electric pulse stimulation. So we give electricity and muscle will contract. Because of these pillars are flexible, if we record with a video analysis, with the microscope, these pillars, are the pillars and this is the muscle, we can measure the force by video analysis. Which force we can do? We can induce what we call twitch force. So we apply a low frequency stimulation, so muscle contract and relax, contract and relax. And this is called twitch force. If we increase the frequency of the stimulation, muscle will not have time to relax, so it will maintain the contraction. This is called tetanic force. So I will have a peak, muscle will maintain its peak till I will release the electric stimulus. So what I'm talking about is this. So contract, relax, contract, relax. This is our twitch force, and you can see here our pillar that is displacing. This is the twitch contraction, and if I apply a higher frequency stimulus, I will have a tetanic contraction where my pillar will displace, its muscle will maintain the contraction, and then when I release the stimulus, it will relax. How we can stimulate several tissues? So with our plates that we created here, that is named MyoMoves. So it is a plate that has several electrodes. These are electrodes. Here is where I put the electric stimulation, and it is adaptable for a 24 multi-well plate. This is the top view. Here are all our tissue, engineered tissue, with electrodes to the side. This is the lateral view. And with this selector, we can stimulate one well by one. This is the single mode stimulation. We can select row by row or column by column, or the wall plate, depending on what we want to do. Okay, what about the tumor model? Because we have functional muscles. So for tumor model, we use collagen hydrogels, where we can, for this first project, we are using pancreatic PANC1 cells that are commercial cell lines. And because pancreatic cancer is one of the more cachectic cancer that there are. So we put our cell in this hydrogel in a single cell mode. And because this hydrogel have some holes, if we let these cells proliferate, they spontaneously form spheroids inside this hydrogel. After 14 days, we'll take the media, the culture media that we will collect to use as conditioned media to induce cachexia. Here is how they appear, these hydrogels. And in a bright field, this is our tiny hydrogel. And here you can see the small spheroids. To be sure that these spheroids were alive, we performed live-death staining. So in green, we have live spheroids, and in red, dead cells. And you can see here that we have very, very few mortality in this system. Okay, we have muscle, we have tumor, so now we have to induce cachexia. This is the protocol, and we let them differentiate, and at day 14, we use 5% of pancreatic conditioning media to put in our engineered muscle. And at the end point, we will perform functional analysis, contraction analysis, structural analysis, and transcriptional analysis. The first experiment is done with cells derived from adult male or of 38 years old. And here we can see that cells treated with conditioned media present lower contraction capacity. If we zoom in this curves, we can see that both twitch and tetanic forces are reduced in cells treated with conditioned media. How they appear, these muscles? So if we stain them, this is a cross-sectional section. Our muscles are full of fibers, and here are the sarcolemma stained with dystrophin, and here the cytoplasm stained with sarcomeric alpha-actinin. And if with ImageJ we can do a mask, we can measure each single fiber to obtain the cross-sectional area, mean cross-sectional area of the fibers inside the wall tissue. By doing this, we have obtained that cells treated with conditioned media presented a higher percentage of fibers with lower cross-sectional area. They are smaller, they have less force, and they are smaller. Okay, we wanted to repeat the same experiment with cells derived from a baby of 14 months, and we obtained almost the same pattern because we had less force in cells treated with conditional media, even if at low frequency we have this strange pattern where it seems that cells cannot relax after the stimulus. We do not know exactly at molecular level what happens, but we have this phenotype. As I think everybody knows, in cachexias, there are some important variables like sex or age that influence the behavior. For example, in this paper, they explain that males are more prone to have higher or more severe cachexia because they lose both lean mass, muscle mass, than fat mass. While female, it seems that they are more protected because they tend to lose fat mass and to preserve muscle mass. So we wanted to repeat this experiment with cells derived from females. And here, surprisingly, we obtained the same phenotype. We took cells from a female of 33 years old, and treating these cells, this tissue, with conditional media, we cannot see differences in the force. So there are sex-specific differential pathways that drive cachexia that are important to be understood. So we have sent these cells and the ones from these tissues and the ones from adult male to do a transcriptomic analysis, and we will let know the results. Okay, as final part, how we can integrate this platform with a sensing platform to obtain good biomarkers, because as Noah said, there are necessity to have biomarkers to understand also the precachectic state, that is the state where we can do something for the patients. So here we can integrate the organ-on-chip technology with sensing platform. Here, for example, we have a media reservoir that is connected to our organ-on-a-chip. This is a really easy organ-on-a-chip because it has 2 chambers with our tissue. Here is an example of microfluidics that goes through the chamber. It's blue to explain, to show, but it should be media. And then after passing to the chamber, it can go to a sensing platform that is formed by a light source and a detector. Here in our group, we use plasmonic biosensors. That a biosensor is this part that is formed from 2 technical replicates. And really short, we use a metal surface that is gold, biofunctionalized with antibodies that recognize and analyze a biomarker. So if the analyte is attached, we will have a different wavelength in the detector, and the shift between not attached and attached, it is measurable. Here, for example, are some testing for with hydrochloric acid at different concentration. So we can measure in real time and quantify well the biomarker. So a conclusion for this first results, this part of results, is that we have developed a fully 3D human model of cancer cachexia with functional muscle assessment. We can reproduce sex-specific differences in cancer-associated cachexia. We have ongoing RNA-seq analysis to identify key molecular pathways. We are planning to do structural and mitochondrial characterization. We are planning also to increase the model complexity, adding, for example, other tissue like adipose, liver, or other tumor types. And of course, it could be a scalable platform for drug testing. We are open to collaboration to increase the complexity of this model. And this is our group in IBEC. We are the Biosensor for Bioengineering group. And here are the principal investigator, Javier Ramon, and Juanma Fernandez Costa. And this is my email if you want to reach us. Thank you.
Thank you so much, Chiara, for this fantastic talk. Please feel free to add your Q&A in the chat. I will start with one. So you show that what you call muscle, and you show a cross-sectional area is pretty big because you have a scale bar of 100 micrometer, correct? So that will be almost a millimeter. How can you guarantee, a perfusion of fluid or nutrient inside it? Because the section and the histology look nice, correct?
Yeah. Yes, we have the perfusion is fine. We have already done for other drug testing or also for nutrients. The tissue is not completely full. We have like some matrix inside, but the perfusion is nice, both for nutrients and also for drugs. They can enter well.
And then I have a last question, then we need to move. But a technical question. You say that these two myotubes are in a tension. How can they attach to the two pillars that are in your system?
Well, they are not completely attached like a tendon. They form like a shape around the pillars because we put the encapsulated the matrigel with the fibrinogen and muscle in the chamber, and at the beginning they are spread. So they will compact due to the matrigel compactation, and then with this tension will direct the aligning of the fibers.
Okay. Thank you so much, Chiara. Thank you so much.
Thank
We
you.
need to go to our last talk of today, and welcome to Abolfazl. Let me go mute here.
Hi, hello. My name is Abolfazl, and I'm from Menzies Lab at the university. We are located at the University of Ottawa, and today I'll be presenting our work titled "Reciprocal Multi-omics Reveals that Modulation of ADP-ribosylation in Skeletal Muscle Shapes LLC-induced cancer cachexia." So I think all of us know that the cancer cachexia is a multifactorial wasting syndrome, which is characterized by ongoing skeletal muscle loss, and often with fat loss, and it cannot be fully reversed by nutritional support alone. It is especially common in advanced cancers and affecting up to 80% of the patients, depending on tumor type and also stage of the cancer. I work with PARP1 or poly-ADP-ribosyl polymerase 1. It's NAD consumer that becomes activated by stimuli such as, like, let's say, inflammation or DNA damage. Once they become activated, it uses NAD to transfer a post-translational modification on the target protein. It can be a mono ADP ribosylation, like a 1 ADP ribose, and it can be a poly ADP ribose. Basically, PARG is the main eraser of this post-translational modification that I told you guys. Hydrolyzes this PARylation and releases free ADP ribose, which can itself act as a signaling molecule. What does PARP1 actually do? At site of DNA damage, PARP, as I said, acts very early in the response. It detects the DNA breaks, then uses NAD and transfer PAR and PARylate itself and target proteins, creates a local signal that helps coordinated DNA repair. This is also the logic behind PARP inhibitors like olaparib. Basically, in a normal cell, even if PARP1 is blocked, BRCA-mediated repair can still help the cell survival. But in BRCA1 or BRCA2 deficient tumor cells, that backup repair pathway is missing. So when PARP1 is inhibited, DNA damage accumulates, and the cells don't have a way to repair their DNA, and they die. So this is the basis of synthetic lethality. So up to this point, I focused on DNA repair role of part 1 because that is what is best known part of the story. But that is also what we may be overlooking actually. Part 1 and parylation regulates a much broader network of stress response biology, including controls transcription, controls apoptosis, controls chromatin remodeling, controls tumor promoted inflammation such as NF-kappa B, and modulate metabolic regulations because it consumes the NAD, and if you change the NAD, you change the metabolism of the cells. So, and then recently, several studies in cancer cachexia and related models suggest that PARP signaling also can influence muscle phenotype and disease progression. So this led us to ask a broader questions beyond DNA repair. How does modulating ADP-ribosylation reshape skeletal muscle biology in cachexia? So we developed muscle-specific PARP1 knockout and using the HSA-CRE-driven systems. And we basically gave tamoxifen to the mice. We got rid of the PARP1 in the muscle, and we looked at the total ADP ribosylation that is on in muscles, and we saw a slight decrease, even though that is not a lot, but it shows that there is a compensation by other PARPs maybe or other poly-ADP ribosylating proteins, but we see a slight decrease. But that changed a lot the proteomics of the proteome of the cells. And when we did transcriptomics, we looked at the gene ontology. We observed that the translation and the mitochondrial gene expression and NADH complex assembly are super downregulated, and among the most downregulated ones. And IL-6 production, TNF superfamily cytokines, and broader inflammatory response, they're upregulated. All of this are super cachexia relevant inflammatory signalings. And we got curious what's going to happen if we keep the mice for a while. When we on the baseline level, on the functional side, we actually did not see an overt baseline phenotype with PARP1 loss. And across these assays, lean mass, grip strength, 4-degree cold challenge, RER using clamps, and also global protein expression assessed by Sunset showed no change. So even though PARP1 ablation reshapes the molecular landscapes that we showed using proteomics and transcriptomics, there's not a lot of functional differences. And then so at baseline they are able to compensate, but the next question we asked was that what's going to happen in stressful situation like cachexia. So we gave the mice, we induced cachexia in the mice, and interestingly, we observed that PARP1 muscle-specific knockout cachectic mice, they quickly lose their grip strength. So you can see it as a green line. And versus it takes longer for wild-type mice to lose their gripping strength, but they eventually end up around like 20% less. And we looked at their tumor-free body mass. They lose their tumor-free body mass equally. But then when we looked at their TA mass or GPS or quad mass, we observed that in cachexia, of course, there's going to be muscle mass loss. But in PARP1 knockout muscles, there is an exacerbated response. So you see here, for example, in TA, there is a statistically significant increase in the muscle mass loss. And you see here in the GPS, again, the same story. And in quad mass, doesn't reach a statistical significance. But we looked at the TA cross-sectional areas and to see how's the minimum for diameters of the muscles. And we observed that overall, the part 1 muscle-specific knockout cachectic ones, they're smaller, even though that their muscle fiber composition did not change. PARG is the major enzyme that removes PARG. So in some ways, it functionally opposes part 1. And PARG inhibition has also emerged as a therapeutical strategy in DNA repair defective cancers, and currently they're in clinical trials. So I basically took this primary myoblasts, differentiate them, and then added PDD, which is a selective PARG inhibitors. And I saw that in just baseline level, and you see that there is the width of the myotubes are bigger. And when I add the conditioned media from LLC cells onto these myotubes, I see, and then I do qPCR, and I see that increase in TRIM63 and then RED1. But when I mix the conditioned media with PDD, the PARG inhibitors, and I add it to the cells, this response does not happen. So we again created a PARG muscle-specific knockout, and you see that the for the gene expressions, PARG expression is reduced in GP and quad again, but not in the liver and IBEC. We took the muscles and then we looked at the transcriptomics and proteomics again, and we observed that the opposing to part 1, translation at synapses, cytoplasmic translations are upregulated in gene ontology assessment. And here again, when looking at the proteomics level, translation at synapses, and then you can see cytochrome complex assembly and then mitochondrial gene expressions is increased. So it's basically opposing response to part 1. So we were thinking, well, what's going to happen if we give the mice cachexia? So when we inoculated LOC onto the flank of the mice, we observed that the again, like cachexia reduces the muscle, reduces the body mass, but the PARG knockout male mice, they're slightly protected, and you see that they are losing less body mass. And here you see on pink, you see the female mice, they are losing the Vol type mice, they are losing their body mass. But when they are cachectic PARG knockouts, you see that there is less body mass loss. When we were looking at the quad, and then we looked at the quad, we looked at TA and other muscles. We observed on the baseline level, like without cachexia, the quad mice, the quad of the mice is actually trending to be bigger in PARG knockout mice. So without any things, they're slightly bigger. And when they get cachexia, it's not reaching statistical significance, but they're slightly bigger too. Here you see the quad in female mice having cachexia. You see there is a quad muscle mass loss. But in PARG knockout, you don't see that anymore. TA muscle, again, you see a trend for increase on PBS without cachexia, but here you see again like a protection, which doesn't reach significance, but it's trending. Then we looked at the cross-sectional area, and we saw that the PARG knockout mice, they are bigger than the Vol type mice. So in one of the biggest hits in my transcriptomics analysis in PARG knockouts, it was myostatin. So myostatin myostatin basically is a central negative regulator of muscle mass. And I basically looked at the part 1 too to see what's happening in cachexia. So you see that in transcriptomics, you see there is an increase in myostatin expression, and you see it again in the part 1 mice as well. But the PARG muscle specific knockouts, you see there is increase in the wild type, but the PARG knockouts, they are not able to increase the expression. And using proteomics, you see again there is a slight trend to increase myostatin. And PARG knockouts are PARP1 knockouts are basically the same as wild types, but when we looked at the PARG mice, you see a decrease in myostatin in PBS alone and also in LLC. So whatever is happening is that the PARG mice are not able to increase the expression of myostatin in response to tumor, and that's potentially protecting them against cachexia. So yeah, so the other thing is that myostatin controls mTOR signaling. Myostatin controls a lot of downstreams proteins that are important for muscle maintenance. You'll see that cachexia, so this is the wild type, PARP1 IMKO, basically PARP1 knockouts, wild type LLC and PARP1 LLC. You'll see in response to cachexia, there is increase in FOXO3, there is increase in TRIM63 and in the UPS components. And then when they get PARP1 muscles get cachexia, they respond like an exacerbated. There is more increase, and it's like which potentially that's why we see an increased muscle mass loss. Here again, the same mRNAs, you can see that cachexia increases it, but interestingly, the PARG IMKOs, they have less FOXO3 compared to the wild type LLC. They have less TRIM63. They have basically less component of the UPS system. The other thing that we looked at was the ribosomal proteins, which are responsible for protein translation. You see that in response to cachexia, there is less ribosomal protein, and when PARP1 mice get cachexia, there is like an exacerbated response toward lower mitochondrial protein, but these things does not happen in PARG muscle specific knockouts, and we think that how these mice are protected against cachexia. And thank you so much.
Thank you very much, Noah, for your very interesting presentation. So again, if someone has answer a question, please just wrote in the Q&A. I have one question for you. I'm start. So did you show that the PARP1 knockout mice have a phenotype in the muscle mass? No, sorry, they have a phenotype in the muscle mass and also in the strength in the early time point, but at the end, no. So what do you think that is the difference between a phenotype in the muscle mass, but not in the strength?
Oh yeah, so that's the thing that we are thinking about. Like we are not totally sure what's happening, and that's the same thing happens with the PARG mice. So PARG mice have increased muscle mass, like they were protected, right? and in response to cachexia, but their grip strength data that we have also shows almost the same level, and we're looking into that.
Do you think that your mechanism here is affecting neuromuscular junction or something similar?
We do see a lot of in our proteomics results. We do see a lot of signals in from neuromuscular junctions proteins, and that's interesting too.
So you show that several data from you measured cross-sectional area in different type of different type of muscle fiber. Did you check if the type of fiber is changing in your model?
Yes, so and I showed the data in my presentation too for the part one. We did not see any changes in response to cachexia and also in response to or knockouts, both part one and part. The muscle fiber types stays the same across all the conditions.
They ask you which is did you check the strength in the tumor mice in your model, because I think you just show in normal mice in basal condition the strength. How is the strength in your tumor mice?
Yes, so I showed for the part one, I showed data that they lose their strength, their grip strength faster than the wall types.
Okay. Yeah, thank you. So, and I think that our time is done, and I just want to ask to the Chiara and Noah to show the face again, so we can really thank you all of three for this fantastic talk, and thank you so much again, and thank you all the attendee of this talk. See you next month. Thank you.